But, oh, there we go. Thanks, Malati. So welcome everyone to this presentation. So this is a presentation from a speaker all the way from Korea, from IVIM Technology. So this technology is an imaging technology that involves a combination of two-photon and confocal microscopy. So 6 different models that they advertise. I won't go through them in detail here, but it is in the advertisement for the MSA Journal Club. So they have various types that depend on your application, your model. So you can get in contact with the salesperson and they can get you more in line with what you would need for your work. But the main purpose is for in vivo real-time imaging and animal models to mimic human response to treatment, pathophysiology, etc., at a cellular level, especially in cancer research. So I know at Northwestern University we're doing some cancer research. So anyone else out there in Africa doing cancer research, this is a good technology to look into. The technology will also work for ex vivo studies involving cell culturing, another good aspect, another good application. So the speaker, Selina, will explain the technology, and thereafter there'll be a virtual demonstration how everything works. So
Yep.
if you're okay, then Selina, on your side, I will hand over to you.
Yeah, sure.
The floor is yours.
Thank you for your kind introduction, Shane. Hi, I'm Selina. I'm an application specialist in IVIM Technology. So it's my great pleasure to meet you, even it is virtual, but very happy to introduce our technology today. So for today's demo, we will briefly introduce technical features of intravital microscopy and in vivo research applications on oncology, immunology, and also metabolic research. And after the brief introduction, we're gonna move on to the live virtual demo to show you how IVIM system actually works in live dorsal Skinfold chamber model with the basic system and software operation. So during the session, if you have any question, please feel free to ask us in the Q&A box or the Q&A session later on. Firstly, I'd like to introduce IVIM's intravital microscopy to observe live tissue and cells in the live animal models. So compared to a conventional microscale imaging technology such as X-ray, CT, MRI, which only provides tissue level and static in vivo information, by the way, the intravital microscopy can directly observe the dynamic cell movements inside of the internal organ of the live animal model for the human disease. So it successfully achieves the dynamic intravital live cell imaging, such as real-time cell trafficking, cell-to-cell interaction, and cell-to-microenvironment interaction inside of the living body. And furthermore, it can be widely applied to the therapeutic research fields, investigating drug delivery to the target's tissue, drug efficacy, and therapeutic mode of action validation in various preclinical animal models for the human disease. So IVIM is the world's first and only one company providing this new all-in-one intravital microsystem with the basic intravital imaging performance. This all-in-one single box package enables easy installation, operation, and maintenance, and it has co-optimized hardware and the software for the best in vivo imaging performance. Well, firstly, ultra-fast imaging speed to capture the very fast cell dynamics inside of the living body. And second, live tissue motion compensation for automatically adjusting the motion artifact for the users to get the high quality and clear images even on the beating heart or lung organ. And lastly, the integrated live animal maintenance system and various imaging window accessories for the wide range in vivo research applications. So with those key in vivo functions together, user can get the high quality and high resolution in vivo data without any limitation in imaging target live organs. So our first key feature is the ultra-fast imaging speed. So all of our system is fully equipped with the ultra-fast laser scanner, the polygon scanning system, which enables the ultra-fast high-speed imaging at the maximum speed of the 50 frames per second with the 512 by 512 pixels each. And also it could achieve the uniform excitation illumination over the entire field of view. Thereby, we can acquire the high quality images and the videos at ultra fast frame rate in real time. Otherwise, the other like competitors' microscopy usually have the galvano scanning system, which only can get uniform excitation due to the unstable scanning speed of the resonance scanner over the entire field, which derives the low quality imaging data with the limited imaging speed as well. Second, IVIM's intravital microscopy system is integrated with the animal motion compensation function. So it enables the automatic hassle-free high-precision motion compensated imaging by facilitating the frame-by-frame motion compensation. So it's the most optimal solution, particularly for the rapid moving organ like heart, as it can compensate the XYZ movement with the real-time acquisition of the compensated imaging data right away. So this is in vivo real-time video of the heart. In this side-by-side comparison, even though the raw imaging data has the high degree of the movement, but when we apply the motion compensation function, we can directly acquire the high quality of the clear image without any shifting artifact. But without this feature, only this blurred image can be obtained due to the motion artifact, which highly disturbs the quantitative cell analysis of the imaging data. So please note that any other conventional standalone microscopy only can acquire these shifted images, like in the middle, from the same animal model. So thereby, only IVIM system has fully optimized the imaging solution for intravital imaging of highly moving live organ in real time. On the third, we are providing various additional accessories for maintaining the healthy condition of the live animal model during the whole experiment. Here we have the rectal-proof sensor for sensing the body core temperature, which has a feedback system with the heating plate at the bottom. So it maintains the stable body temperature throughout the whole experiment. And also there's a glass heater for the local tissue temperature maintenance of the imaging target organ. Therefore, we can keep the stable status of the both of the body core temperature and also the local tissue temperature of the imaging target organ simultaneously at once. So this is the translational stage for mounting the mouse model. So every accessories are connected to the connector in the left, and we can slide in and out of the stage before and after mounting. And moreover, the system is fully compatible with the gas anesthesia system through the tube adapters in the side wall of this box. And inside of the imaging chamber, it is fully equipped with the live animal monitoring system for the real-time monitoring of the animal status during the whole imaging experiment. And finally, we are providing these in vivo imaging window chambers that are fully optimized for the target organ-specific imaging applications, especially for the long-term monitoring. Dorsal Skinfold chamber is for in vivo research of the cancer or drug candidate. And for intravital heart and lung imaging, we utilize the suction-based tissue motion stabilizer window chamber system to temporarily stabilize the imaging side of the beating heart or lung tissue. And for brain research, we implant the cranial imaging window in the skull to visualize the brain cortex in a long-term manner. And abdominal imaging window is for abdominal internal organ imaging, such as pancreas, spleen, kidney, colon, etc. And for long-term monitoring of the mammary tissue and the pancreas, we can utilize the mammary imaging specific window chamber and pancreas imaging window chamber together. And for the uterus imaging window chamber is also integrated with the suction-based stabilized system for stable in vivo uterus imaging. All of those are fully optimized and provided as the accessory product of IVIM system. And moreover, we are providing in vivo labeling kits for various interest target cell or the molecules inside of the live animal model. So it includes the target cell-specific antibody dye conjugates, such as CD11B or CD45 for the leukocytes in vivo labeling, or Ly6G or Ly6C for neutrophil or monocyte labeling, and F480 for the macrophages, and CD31, the PCAM antibody and dye conjugate for the vascular endothelial cell labeling in vivo. So these have already been optimized for the optimal concentration and the dosage for the high level of repeatability, reproducibility, and high reliability for in vivo imaging research. So just by IV injection to the mouse model, then 2 hours later, the clear fluorescence signal on the specific target cell is coming out in vivo. So users don't need any additional optimization for in vivo trials. So through this intravital imaging platform, user can do various preclinical in vivo studies, including analyzing in vivo immune responses in the live animal model, tumor model, or various human disease animal models. And also we can utilize this intravital microscopic imaging technology to monitor the drug delivery, drug distribution to the target organ, and also we can perform in vivo analysis of the drug efficacy evaluation in target skin or the target tumor tissue. Finally, IVIM's intravital microscopy has succeeded in high-quality live cell imaging in almost all of internal organ in a live animal model. So it includes some brain tumor, skin, lung, pancreas, spleen, and GI tracts, prostate, lymph node, memory tissue, and muscle, and even heart and lung, and retina in vivo. And from this slide, I'd like to highlight the in vivo research applications by the target research proposed. So this is the case study evaluating the drug delivery in the liver. So we validate in vivo nanoparticle drug delivery and accumulation of the drug was recorded with the real-time video in a sinusoidal structure. The red nanoparticle drug were delivered to the liver sinusoid right after IV injection by the tail vein catheter, and then it gradually accumulated to the specific sites of the sinusoidal vessel over time. In the disease model, especially in the NAV field, fatty liver disease model, we can clearly visualize the fibrogenic collagen and the lipid drumlet deposition in the liver tissue. Especially the fibrogenic collagen can be visualized with the label-free second harmonic generation signals in a two-photon microscopy. Also, here's the imaging result of monitoring the development of the hepatic lipid drumlet in the non-alcoholic fatty liver disease mouse model. So we visualize the disease development over time. So the number of the lipid drumlets accumulated in the hepatocyte increased over 3 weeks in the liver disease animal model. And also we can perform quantitative analysis of the lipid drumlet size and area near the various type of the hepatic vessels, such as portal vein, periportal, and sinusoidal vessels. So in the result, the size and the area of the lipid drumlet increases from the portal vein to the sinusoidal vessel during the non-alcoholic fatty liver disease development over time. In the steatosis disease model induced with the 27 weeks of the high-fat diet, the hepatic lipid drumlet and the fibrogenic collagen were accumulated in the hepatic tissue, especially the significant fibrogenic collagen deposition in the liver stroma, which means the liver fibrosis was successfully visualized by the label-free second harmonic generation imaging. Compared to the sham control model, the hepatic lipid drumlet and the perisinusoidal collagen deposition were significantly observed around the hepatocyte in the liver of the steatosis animal model. So, and in the magnified 3D rendered data, distinctive feature of the advanced NAV field, such as accumulation of the lipid drumlets and also perisinusoidal deposition of the collagen structures were clearly identifiable. And furthermore, we could precisely quantify the volumetric proportion occupied by this hepatic lipid drumlet and the collagen fibers in the liver tissue in vivo. So in this way, we can introduce an innovative research perspective to comprehend the various cellular mechanisms within the context of the diverse metabolic disorders. In a lung of acute sepsis model, we can observe the dynamic inflammatory cell responses, such as pulmonary neutrophil migrated to the specific sites and block the microcirculation of the lung blood vessels. By the real-time imaging analysis of the blood flow, we could identify the functional capillaries and analyze the blood flow velocity in pulmonary microvasculature in the sepsis animal model. Next is for long-term monitoring of the target organ. We generally utilize the imaging window chambers, for instance, the dorsal Skinfold chamber for in vivo cancer study. So this is the surgical procedure for dorsal Skinfold chamber mouse model. So implant the imaging window chamber on dorsal skin and clip the three sides of the chamber and fix the screw and make a suture and remove the one side of the skin tissue. And then we subcutaneously inject the tumor cell in the other side of the skin tissue and cover with the cover glass and firmly fix it well. By using this cover glass clip, and we could finally perform the repetitive intravital imaging of the tumor development and in vivo analysis of the target drug delivery in the tumor tissue. Therefore, intravital cell analysis of the angiogenesis, vessel dilation, or the blood flow using RBC dye are successfully acquired. In this study, we will evaluate the anti-angiogenic drugs efficacy on the tumor mouse model. So firstly, we implant the tumor cell in the same way on the sham control model and also the anti-angiogenic drug treated group and start the monitoring from the 7th day after the tumor implantation. And visualizing together with the tumor vessel, we can analyze the drug's anti-angiogenic effects on the tumor tissue. We could see that in the drug-treated group, the tumor vessels are less dilated compared to the normal sham control group. And in the wide area after the drug treatment, vessel morphology became more normalized, that is less dilated and less branched, and compared to the sham control model. So in this way, we can finally validate the in vivo efficacy of the anti-angiogenic drug on the tumor vessels. To summarize the research application on oncologic research, various tumor models, including some primary tumor, metastatic cancer in different tissue or organs, can be analyzed in vivo for the drug delivery inside of the tumor tissue, and anti-cancer drug efficacy, such as the tumor apoptosis, vascular changes in diameter, density, and vascular permeability, and also the blood flow, can be analyzed in vivo. And also we can quantify some immune cell dynamics and cancer metastasis in the live animal model. We have published a number of intravital imaging research publications by using our system, categorized by the imaging organ and imaging purpose, such as the in vivo analysis of the microcirculation, drug delivery, and cell transplantation. So those could be a great source for your imaging applications. And here are our researchers worldwide. We are actively supporting the researchers by providing the optimal intravital imaging solutions, which meets their research interest with a strong academic background. Next, we are gonna quickly jump into the virtual demo session. Today, we will perform in vivo dorsal Skinfold chamber mouse model with the basic system operation. So we're gonna use both of the confocal and two-photon dual mode microscopy system, which is IVIM CM3. And for the software, there are two types of software. The IVIM engine is the imaging acquiring software, which is fully optimized for the real-time in vivo data acquisition. And IVIM Studio is the post-processing software for the 2D to 4D visualization and analysis. So Luna will soon show you the basic system operation using those two softwares by acquiring the mosaic and 3D Z-stack imaging together. And lastly, the animal model for the liver imaging will be the CX3CR1 GFP transgenic mouse model, which is expressing GFP in monocyte and the macrophages. And moreover, will additionally label the blood by IV injection of the Evans blue. So our applicational specialist, Luna, has just prepared the experiment for the virtual demo session. Hi, Luna. Would you start the session, please?
Yes. So, hello, this is Nuna, Field Application Specialist from IVIM Technology. So today I'm gonna show you a video intravital imaging of back skin of a dorsal skin for the chamber implanted mouse by using IVM CM model with the data. Thank you. So as for the beginning, I'd like to introduce the convertible confocal and the two-photon microscope IVIM CM3. User can easily recognize the main body of the CM3, which has such a compact design with powerful performance compared to other devices. And below it, there are laser source for confocal and the two-photon. In this imaging room, an automatically rotating toilet for 5 objective lens is furnished on the ceiling. And underneath, there is a stage controller controlled by jog dial. And we can see the 5 connector already attached to the retoporb and the preheater. So here you can see this is our animal maintenance system with bi-directional feedback friction by getting information in between the retoporb and the heating plates, so that research can keep stable temperature of the whole body of the mice. So when it comes to the surgery procedure, to the implantation of the dorsal skin for the chamber onto the mice, I will briefly explain it. After we've gently folded the dorsal skin, one side of the back skin was removed in a circular way and replaced with the 12mm cover slip. Then, dorsal skin for the chamber implanted mouse model was mounted on the holder for the long-term imaging, as you can see on the screen. So, to start the imaging, I will slide the animal stage inside and close the door. So now, let us jump into the imaging section with the CM3 microscope. Screen of the CM3 microscope will be shared for you in a second. So what you are seeing now in the share screen is IVIM engine and imaging acquiring software for IVM CM3 model. When you closely looking at the control panel on the left side, user interface seems to be quite user-friendly. And on the right side, on the first row of the main screen, there are the main display for the averaging adjusted video. And then we have the live video. Then of course have the window for the capture imaging. Underneath them, colors separated display for the top P, GFP, RP, and the Sci-Fi channel would lie along the second row. So in order to turn on the microscope on, I will make the scanner on. Next, adjustment has already done for each laser power and the detector value for excitation and the detection for this in vivo imaging. Lastly, when I turn on the shutter on and find the focal plane until the signal are on the display. So here, as you can see, we can see the blood vessel from the Sci-Fi, which labeled with the advanced blue. So user can use the drug dial, which get us to navigate the best of view of this cover glass. So those large blood vessels like this on the screen with the several branches along with the small vasculature may seem to be good spots to mark and take a signal from. So again, concerning the fluorescence labeling, advanced blue was tailband injected before the imaging in order that we can visualize those blood lumen on the Sci-Fi channel with the blue color. With the red color. And here we use the CS3 CL1 GFP transgenic mice to follow the monocytes and the macrophage in the green color. So in IVIM engine, it makes it possible to imaging a number of the combinations such G-state for death wave imaging, mosaic for the wider area imaging, multiple position for multiple spots, and we also have the time lapse for the temporal interval, as well as live video recording. As for this imaging section, I'd like to tackle imaging with the combination of the G-state with mosaic mode from the region of the interest. In order to perform wide area imaging with the IVIM microscope, X and the Y coordinates of the field of view from the first start position would be loaded. Then we need to load this the final position of the mosaic. And here we need to design the overlap of the each images. So we will do the 2 by 3, totally 6 imaging for the mosaic. Additionally, on the same field of view, coordinates of the Z would be loaded from the surface to the bottom with the interval of 5 micrometer from top to the bottom. So the total imaging path would be like 100 micrometer. Once it attacks, we can have an imaging like this. So during the imaging acquiring process of the G-state with the mosaic, I'd like to share those imaging data that has been already taken in this morning. Experimental design is somewhat a little different, but good to show immune cell dynamics. On the left side of the PowerPoint, it's a 5x4 mosaic images in the width and the length way of the skin. On the right side, there comes the imaging of the first spots here, acquired with the 10 times of objective length. Furthermore, if research makes a choice for the better resolution of objective length, such as 25 times water immersion length, such crystal clear imaging at the cellular level can be obtained without any delay. So I hope you could understand those limitations of us not being able to show every function in this short demonstration. And now I'd like to show you the IVIM Studio, which will be on the screen, share screen now, which we can use the IVIM Studio to do the post-processing. So this is the 2D visualization mode of the IVIM Studio for not only 2D post-processing, but also 3D and 4D in vivo data analysis with the user-friendly UI. From now on, with the one of the single taken imaging of the mosaic, I will change the adjustments of each color balance like this. And once the adjustment be done, nextly a set of the G-state imaging will be played with the G-state label. And we also can insert the scale bar here. Then we can see what difference from each layers like this. And it's completely possible to perform best functions such as making the position. Like this. And furthermore, quite time-consuming and demanding one like making mosaic. So we can stitch the imaging together like this. In this final chapter of our demonstration, so now I want to show you the 3D visualization mode from the G-state data. This is a 3D visualization mode of the IVIM Studio. Powerful tools for the 3D and 4D in vivo data analysis. That way taken sequence would be used for the 3D reconstruction. So again, we can make some adjustments of this imaging. Then we can recode the 3D rotation polarized way. So by this way, then we can realize the total structure of the blood vessel of the back skin. So thank you for your listening. So if you have any question, just let us know. Thank you.
Thanks for the wonderful demonstration, Luna. So next, we'd love to move on to the Q&A session. So if you have any question according to the Intravital applications, please feel free to ask them now.
So are there any questions in the audience? You're welcome to raise your hand if you want to ask verbally, or you can write it in the chat box if your mic is not working. So while we wait for questions, oh, there we have Anisha.
Hi. Yes, I was just wondering if you've noticed any interference, metabolic or immunologic reaction to the dyes that you use in vivo. So at the conference, actually at the immunological conference in December, I spoke to a girl who's doing research on the dyes that they're using, and they actually found that it leads to an immunological response, and this kind of interferes with some of their results.
Oh, thank you for the question. Yeah, it is possible, but we always like set our experimental design by using the same control model, sham control model with the experimental group together. So we can like control that kind of interference. But yeah, in case of some like antibody binding can like cause some a blockage of the cell to cell interaction actually. So yeah, it is possible. But and also the in vivo labeling probe that we are using is already optimized by the publications already. But yeah, it still has some like possibility of the issues that you mentioned. But yeah, to avoid those kind of like interference, we always make the control model together. Yeah, thank you for your like question.
Anisha, do you have any other questions?
No, I think that's it from me. It looks really cool. I think this is a great technology and will have many applications in the future for research and clinically.
Okay, thank you, Anisha. Anyone else have any questions they'd like to ask our speakers? I have a question regarding the quantitative side. You mentioned that you can quantitate certain aspects. How do you do that with a microscopy? I might have missed that part.
Oh, it is a kind of like image analysis. So for example, when we like get the time-lapse imaging, we can get some like cellular movement, cellular moving like videos from the time-lapse imaging. So from that video data, we can like track each individual cell over time. So that from that kind of like analysis, we can like measure some like cell velocity over time, or we can count some cell-to-cell interaction contact, or we can also like measure the vessel diameter changes, etc., from the imaging data.
Okay. So yeah, so it's pretty much extrapolation from your qualitative images that you're seeing, your high resolution qualitative imaging.
Oh, thank you very much.
Have you guys I saw the application of is also with cell cultures. Have you done any projects yourselves with the cell cultures?
You mean in vitro cell cultures?
Yes.
Yeah, it is also compatible with the in vitro cell culture imaging when we just optimize the stage accessories for like cell culture dish. But basically, our system is like specialized system for the live animal imaging, but still it is it can be like compatible with the in vitro cell culture imaging and also ex vivo tissue slice sample imaging as well.
Okay. Yeah, I
Hi,
would be
Prof.
very
Mason. Sorry, it's Dr. Barke. I'm one of the sales reps for Africa for the IVIM equipment as well. So I just
sure.
if I can just step in as well. Like Selina said, the system is designed specifically, or not designed, but optimized for live animal imaging, and that's its main advantage. But I find that the whole advantage in our market, specifically with our universities, is that you want to equipment that can be diverse and can be used across multiple disciplines. So it can do ex vivo imaging, and then it can then be expanded onto intravital live animal imaging, and it has multiple research disciplines. So it can do cardiac, it can do lung tissue, metabolic. So it can do all these sort of studies. So we're trying to really push it as a versatile equipment, because I sort of know the situation and how we're dealing that. If you're doing a large equipment funding, you don't want something that's tailored to just one use. So we try to push it as being a very versatile tool.
So I'm glad you came out the dark there, Ben, and spoke. So Ben is the local distributor for IVIM, if I understand correctly.
Yes, yeah.
So Ben, would you be able to supply your contact details, perhaps in the chat or something like that?
Sure thing.
So if anyone wants to have perhaps more information on the products, you can contact Ben directly. And Ben is in contact with IVIM in Korea. And he can certainly try and get any questions answered. So yeah, part of the Journal Club, we encourage any listeners to contact the speakers. So we put the presentation onto our YouTube channel, Metabolomics South Africa YouTube channel. So anyone who is unable to attend this session can always view it later. So anyone who can't see the chat box in the YouTube world, you can go to www.benny.co.za, so benny.co.za, and the email is sales@benny.co.za. Ben will be the person who will then receive this email. And if you're interested in this software, hardware, or anything similar to this, I would encourage you to contact Ben.
I have another question, if that's all right.
Go for it, Anisha.
You mentioned metabolic applications. I was wondering if you could just expand on that a little bit. What kind of metabolic applications were you referring to? And how do what kind of dye do you use to look at that?
Actually, we have experienced two types of like metabolic research. The first case I already like shown in the presentation, like visualizing some hepatolipid disease animal model by analysis of the lipid drumlate and fibrogenic collagen deposition over time. And the another one might be the microbiome distribution in the small intestine. So we label the microbiome and we adaptively transfer it, and then we see what's happening inside of the small intestinal villi together with the microbiome together. So that might be the like intriguing research application in Vivo on the metabolism research.
Yeah, that sounds very interesting. Thank you.
Thank you very much.
Yeah, in South Africa, we have a lot of infectious diseases research. So my interest would be then to see how this imaging can be used to determine the immune response to infectious agents such as tuberculosis or HIV. So how the immune cells respond in a particular organ. So in case of pulmonary tuberculosis, it affects the lungs. So what are the first responding immune cells versus the progression of the immune response over time? Have you done anything like this in the past with infectious diseases?
Oh, well, yeah, we infect the mouse model by the salmonella and see as I remember, the small intestine, and then we see some neutrophils in the monocytes infiltration inside of the small intestinal wall during the like infection response over time. So the immune response might be the like great target to like visualize in vivo, because we can see such a quite like fast dynamics over time, which might be the intriguing like data for like presenting and evaluating the some like disease models development, or we can like we can say which target cell it can be affect the which cell, and it can like finally result some kind of like molecular mechanisms in a research, like something that kind of like story can be like available through by using IVIM system.
Yeah, I think there's a lot of application there, so I'll definitely share with my colleagues in the infectious diseases field, not just the pathogenesis of the disease, but also as drug delivery and efficacy and the actual tissue and the response to that. So this is something we probably want to look at as well.
Yeah. So drug
Yeah,
discovery research might be the good, target as well.
I just
Sorry.
also want to put inputs just doing infectious disease, and especially infectious disease in South Africa is really synonymous with HIV and TB, among other things, but those are two main driving factors. And most of the research that comes out normally originates within South Africa. So any sort of supporting literature or anything that is associated with specific equipment is a little bit scant in terms of TB and HIV. So we tend to look at other organisms that have a similar reaction to that, and then sort of extrapolate from there. So regarding TB and looking sort of at the initial infection and primary and first reactivation and looking at sort of latent and active TB, it's definitely something that's possible with the system, because you can do in vitro lung imaging. It's a little bit difficult to do longitudinal studies in those specimens just based on the invasiveness on the operation. So we tend to do either we'll have to have different mouse models at different stages, and then you can visualize the mouse, different mouses at different stages of infection and look at images of that. But in terms of doing longitudinal studies, it's a little bit more difficult, but it's more favorable in terms of the other diseases like HIV, where if you want to monitor vasculitis or specific region, then you can do a longitudinal study there. And then with cancers, it's really easy to do longitudinal studies based on the dorsal skin chamber.
Yeah, there's quite a lot of applications. Yeah, I'll definitely share with my colleagues in that area. I hope they watch this video and have some ideas, and maybe they can throw some ideas at you, Ben.
Yes, perfect. I'm always ready for knowing what they're interested in. And then because IVIM, we also try to help tailor research specifically to the unit. So if anyone has any ideas or anything like that, we try to adapt. I know, for example, other institutions are looking at using the system for zebrafish because of sort of their fast reproducibility rate and sort of very easy to visualize in utero. So, I mean, we adapt the system where we can, and also for specific types of research.
Okay. I know there's also another group within a department that does research in nematodes, C. elegans. Have you done any imaging on that sort of capacity?
I don't, as far as I know, we try to research a little bit for the angelfish. We know it's possible doing it with confocal and multiphoton imaging and doing it on live specimens. Selina, do you know if you've done anything on nematodes specifically? I think the system would be able to be adapted for that. Just want your input.
Yeah, not yet. We haven't like get the great data yet, but it is quite like possible to visualize the zebrafish as well, because the zebrafish is quite transparent compared to the mouse model, you know. So yeah, we expect it is like perfect target as well. So no matter what, we can like visualize it in a good quality.
Okay, so there are a lot of applications that has been done in the past and haven't been done yet. Maybe we can explore some new territory in that area. And I hope that there are some other people in the audience or in YouTube land that are having ideas of how to use this technology. So thank you very much to Selina, Luna, and Ben for all your inputs, for presenting to us. We really appreciate it.
Thank you very much for having us, and thank you very much for your taking time today. Thank you.
Thanks so much. And I encourage anyone in the audience to contact the speakers. I'm sure they'll be happy to hear any communications. I will certainly in the future contact them and see how we can take this forward.
Sure,
Okay.
I'm very looking forward to it.
Perfect. Thank you.
Thank you.
Okay. Thanks so much.